Hi guys, this is Aubrey from SyntheoGenesis, and in today's video I'm going to demonstrate how you can purify a plasmid DNA, circular plasmid DNA from bacterial culture. It's MRFP1 plasmid DNA that expresses the red protein, and in the next video that I'm going to do after this, in a kind of like couple of days later, I'm going to transfect the yeast cells to express this red protein. So it's gonna be fun because transfecting yeast cells opens a lot of opportunities for the researcher, because it is used in basically everything, starting from like brewing beer or in medical purposes, or even making some bread. So hope you like this video, and let's make it work. The process of Miniprep is rather straightforward. It is done step by step according to protocol provided by the scientific facility or by a company. But the problem is that some of the protocols don't include an overall analysis and deep dive into a purpose of specific chemicals and buffers used in this protocol. So for many people, the purification of plasmid DNA has become blindfolded and tedious process, a sheer routine. And this is the purpose of this video, to help Miniprep make little more sense. In order to purify some bacterial plasmid, we will need a live culture of this specific bacteria. E. coli is a literal coffee machine of plasmid DNA, so by culturing an E. coli into an Uppbeat, we basically duplicate dozens of plasmids in it. The key here is that we use a fresh culture, so one day grown culture is just enough. Make an Uppbeat medium and add an E. coli strain in it, and let it grow by the mighty power of mitosis for a day. Now I'm gonna use an autoclave to sterilize my LB media in order to eliminate all the possible contamination in my culture. Now, after the sterilization, take all the glassware and LB media out and wait for it to cool down a little. The sole reason that we're waiting for it to cool down is that we're going to add an antibiotic stock solution to it. That is just a way of selecting for an E. coli that have a plasmid and killing off literally everything else. After cooling the media down, add an antibiotic stock solution. In my case, it's 10 microliters of ampicillin. And now add a bacterial culture in it and let it grow for 1 day. The next day, you're gonna have a bacteria sufficiently multiplied into the culture tubes. And now we can finally get into a Miniprep procedure. The first step is to transfer overnight culture into a microcentrifuge tubes and centrifuge them onto a maximum speed. For me, it's 10,000 RPM for approximately 2 minutes. After the centrifugation, you will be able to detect a cell pellet into a tube. Pour out the supernatant and add 100 microliters of resuspension solution to the pellet, which contains RNase that precipitates the RNA molecule into a cells. Now, mix well with inverting tubes up and down and incubate it for approximately 1 minute. Now you can add 1 microliter of visualized solution. Addition of visualize is optional but pretty beneficial because it stabilizes the lysis process during the Miniprep. Now we can add 200 microliters of lysis buffer and mix the solution by inverting the tube. After adding the lysis buffer, the solution will turn blue. This is an indication that the reaction is progressing in a right pace. If the solution doesn't turn blue, you might assume that there is something wrong with the reagents or stock solution itself. You can now add 350 microliters of neutralization buffer. After addition of buffer, the blue solution will turn white. You can now centrifuge solution on a maximum speed of your centrifuge. Throw away the pellet and transfer supernatant into a sterilized EZ10 tubes. By adding this solution to the silicon membrane, you basically allow plasmid to bind to the silicon membrane because of the high concentration of the salt solution. And everything else, including proteins and cellular debris, will pass through the membrane. After that, add 750 microliters of wash solution that also contains an ethanol. The ethanol into a wash solution helps salts to get more dissolved into our stock solution. Furthermore, the DNA stays untouched. That amplifies the purity of our final result. For the higher yield, you should repeat the wash step once more. After the wash step, transfer the silica filtered column into a sterile tubes and add 50 microliters of elution buffer, and you can now centrifuge it on 10,000 RPM for approximately 2 minutes. Congratulations, you successfully purified bacterial non-chromosomal circular DNA from a live bacterial culture. The next step for me will be to store that DNA and use it in yeast transformation protocol. Yeast transformation is rather hard compared to a bacterial transformation because it is an eukaryotic organism, and eukaryotic organisms are generally a little harder to engineer. But it's doable, nothing really hard, and I'll probably need to swap out the resistance region because this plasmid is using an ampicillin resistance region, which I can't really use on yeast engineering, so it might take a little longer than expected. But hope you like this video. I would love if you'd be able to share this video to your friends and peers, like this video, and just generally subscribe the channel for more updates and more interesting videos like this. Thank you and see you next time.